All right. Thanks, Mike. Um, yeah, as mentioned, I'm going to start it off and then I'll pass it over to Dimitri. So we listed a whole bunch of people here, mainly because of all the research updates we had. We want to kind of just quickly touch upon all the different projects going on with lake trout. So I'm going to start out talking about the status, largely based on the gillnet survey we do with the collaboration with DEC. Oh, okay. All right, cool. Well, while that's loading, I'll just start talking about the survey in general. So it's the the data I'm going to be talking about is mainly on the U.S. side. It's been going back since 1983, and currently this year we're hoping to all set. Okay, to try to optimize the survey design and try some new things. So lake trout under the fish community objectives or under fish community objectives 3 for the deep pelagic and offshore benthic goal. And also with the gillnet survey, we can index a couple other cold water species, burbot and lake whitefish. Lake trout also in Lake Ontario have a binational management strategy for lake trout restoration. So some of the indicators that are specified in the report come from the data we generate in the gillnet survey. And recently in 2022, the Lake Trout Working Group developed this research priorities document to help guide some of the future research efforts, mainly focused on early life history. So this is kind of the general geographic areas where the lake trout survey fishes. It's distributed across 14 different zones, and in each zone, the location of the coordinates are randomized. You'll see the typical, effort with any gillnet is usually one night fish, but I threw in a couple of these points that have 2 to 3 nights because we encountered some pretty strong currents that we were unable to get our gear back the next day, but I still wanted to include some of the data from them. I'll say why at the end of the talk. But just including the nets that were fished for one night, those are used in the trends for abundance analyses in general. We caught 418 lake trout this year. About 83% of them were stocked fish with coded wire tags. Another 14% were stocked fish without coded wire tags. And then 2% were wild fish without any clips or coded wire tags. Here's just the illustration from the surface current maps. That was the reason why we went back out to go find our buoys, and we could not find them. They were towed under for a little bit. This is jumping into the total catch per unit effort of lake trout, broken up by maturity, males and females. And the number for the abundance for 2025 was 10.5 fish per lift, which is one of the lowest values we've seen since 2009, which is okay when you consider the changes in stocking that have taken place. But it's driven mostly by a lower number of immature fish compared to the last 10 years. Here's a graph of stocking levels on the US side and the mix of strains used. So I mentioned there's been a reduction in stocking. That was in 2018 and then again in 2020. And the recent numbers on the US side have been about 320,000 fish stocked. So in 2025, it's a pretty even distribution. This is just the composition of the different strains we see based on coded wire tags. 26% were from the Superior Klondike Reef strain, 22 Seneca, and 22% from Lake Champlain. We're also starting to see Perry Sounds showing up. We've been seeing them for a few years, but they were first stocked in 2015. Maximum age in the survey has been sort of steadily climbing through time. The highest point was, I think, in 2021. We got a 37-year-old fish. This year, the highest was 30. Wild produced fish. This is broken up by immature fish on the top and mature fish on the bottom. The indicator that's specified in the management, one of the indicators for wild fish is that they want to see notable increases in wild abundance compared to the 1994 to 2001 period. So looking at the top one, I was like, all right, well, there seems to be some significance there. So I took the 2020 to 2025 average, which is about 2.2 fish per lift, and that was significantly greater than the average during that time period. However, if you look just since 2022 to present, there's kind of seems like it's been dropping a little bit. This past year was pretty low, and there's no difference in the wild mature fish over that time. So there still seems to be some sort of bottleneck between some of the immature wild fish we're seeing and not making it to adult stages in the gillnet survey. Wild fish have also been detected in both April and fall bottom trawls we have. So the graph on the left here is from the spring prey fish survey. There's been sort of an increase, but starting in 2015 was also when we started to sample a whole bunch of new areas where they likely occur as well. So this graph over here highlights some of the catches of, especially, it's really hard to see. I'm sorry, this is a length frequency index broken up by a different area. And the past 2 years, there's been some age zero, presumably age zero, lake trout detected from the Adolphus Reach area. So that's pretty exciting. Another indicator in the plan that it was intended to show a level of natural or a suggested number of abundance of females that would allow natural reproduction to occur is this catch per unit effort of mature females greater than 4 kilograms. And you can see for most of the time period, especially in the past decade or so, it's the indicator has been largely above the target level. Started to take a slight decline this past year. It was like 1.97, so rounded, it's right at the target level. But despite this trend being above the indicator for most of these years, you're still not seeing that significant natural reproduction that's called for up here. Lake trout condition index through this program continues to be high. This is the predicted weight of a 700 millimeter fish. You can see it's increasing over time, especially in this more recent period. And we think, we've done a strain specific analysis, and this seems to be driven in part by the Klondike Reef strain, which is one of the more common strains we catch now. It has a higher body weight at a given length compared to the Seneca and Lake Champlain strain. And this is the percent composition of adult spawners age 6 and above based on the coded wire tags, just going back to 2000. And I just wanted to highlight here that so this would be the adult spawning stock that would be potentially producing offspring. And throughout the 2000 to 2010 period, you can see it was largely dominated by yellow here, which is the Seneca Lake strain. But now we have a sort of different adult population out there in the more recent areas, where superior Klondike Reef strain are, almost 30% here. So there's more diversity in the spawning stock of these different strains. We're also starting to see Huron Perry Sound show up as adult spawners potentially. But superior Klondike Reef is one that's no longer being stocked. So going forward, this will probably start to change a little bit as they phase out. Adult survival, the dashed line here is the management target of 60%. This is adult survival between 7 to 11. For the most part of the time series, its survival's been pretty good. It's been above that target, especially for Seneca and Lake Champlain fish. They tend to outperform the other strains. Sea lamprey wounding has also been pretty good. It's been exceptional. I think for Lake Ontario, it's been at or below the target for most of the time period. And in particular, this past year, out of 407 fish that were in the specific size category, we didn't detect a single A1 wound. And I think it should be the first time ever on this graph. Just real quick, a couple other species of interest here. Like I said, you can index burbot from this program too, and some of these other cold water species. I've often looked at this burbot graph compared to what the Lake Erie cold water task group generates, and it seems pretty similar to me, although this part's different here. We haven't caught a burbot on the U.S. side in the past 5 years, and that's a new kind of pattern. It seems to be emerging, but Cisco, on the other hand, tend to be a little bit more common now in the survey, although they're overall pretty rare. But primarily still coming from catches in the Eastern Basin. Lake whitefish, we had a couple peaks, one in the mid to late 90s, and then, but overall, it's not really showing much of a trend. We tend to catch them again primarily in the Eastern Basin in this survey, but occasionally by Oswego too, and that fits with what I've seen from the telemetry work, where they seem to be moving down to Oswego, but kind of staying in the Eastern Basin. Last but not least, this was the first time since 1983 that we caught a bloater in this survey. And this, I had to include this because this was one of the nets that we left out for 3 nights. And, you know, we traveled all across the lake to go back to the site to get the net, just to get the gear back. And lo and behold, there was a bloater in it. So that was exciting to us. Thanks to Dr. Amanda Atkins and her team for confirming our identity and, on this one with genetics. It was also a female, so you can see the egg mass up there. So yeah, that was pretty exciting for us. And now I'll pass it over to Dr. Gorski.
All right, thank you. Good morning. I have the pleasure to provide an update from the Lake Trout Working Group on a lot of projects, great work that all our partners are working on. So as Brian O'Malley said, you know, we have this restoration research priorities document from 2022. Partners got together and identified 4 major research focuses that we should be working on. Location of spawning areas, improving life stage sampling, understanding what impediments limit early survival, and continuing working on measuring population dynamics. Over the next few minutes, I'll walk through where we stand on each of these priorities and what's coming next. And so the way I laid out my update here is I'll take each priority, I'll talk about what projects are ongoing and what might be coming up in the future. And when possible, I have a highlight slide from the partners' work. And so for our first research priority, we've made a really strong progress. Last year, I believe Lucas presented an update on the lake-wide substrate imagery project that we completed. So that project's been using drop camera imagery to identify the substrate of possible spawning areas across the lake. This was a lake-wide effort, included lots of agencies and a lot of work. And so that manuscript is currently underway. Coming up after the break, Alex Gatch is going to be presenting an update on our lake-wide acoustic assessment. And so in short, we've been using acoustic telemetry to understand where lake trout are being detected during the spawning period. Our next steps, we're hoping to deploy some fine-scale acoustic arrays once we've identified spawning areas of interest. And so that will be looking for site-specific quality characteristics of the spawning habitat that lake trout are using. Additionally, we are working, Fish and Wildlife, USGS, and NOAA are working to do bathymetry mapping, characterizing some of these substrates and real fine-scale habitat quality. For research priority 2, we've begun evaluating tools and techniques to collect eggs and larvae in a standardized, repeatable way. Our goal is to incorporate this as a survey to do a lake-wide assessment of how egg deposition is happening and larval emergence. And so suction pumping and diver-assisted trapping have both been piloted and assessed. We are working towards coming together this spring to discuss how to evaluate those comparatively. How do we do it across different habitat types and under different conditions? So that's our next step, is to get this meeting to take place. And then we're also hoping to standardize what sort of environmental measures we're taking there, whether there's, flow, temperature, DO, wave energy, things like that. So here's a highlight from the egg suction pumping. Lindsay Colas has been working on out of USGS Oswego. Using this suction pump, they lower this down on the habitat and try to suction the eggs out of the substrate. What they've been able to show is were able to collect embryos between 3 and 6 meters deep. The pump also works well across a variety of substrates. So this is just a great example of the work being done within the lake trout working group, and shows one of the possible ways to quantify egg densities and assess spawning across different substrates. So research priority 3 is actually, you know, limited in what we can do, because a lot of the work we want to do, we need to know these really site-specific characteristics of spawning habitat. But where possible, projects looking at impediments to early survival were begun and carried out, that didn't rely on that knowledge. And so you can see here, there's quite a few. We've completed a strain assessment to understand what strains of lake trout are producing offspring, and we've been looking at thymin levels across strains. And so the thymin, the genetic work is being worked up. Manuscript's going to be in preps shortly, and the strain-specific thymin work was just published recently. Ongoing work includes testing embryo survival on different substrates in the lab, and then additionally looking at prey quality and how that might influence thymin level in adult lake trout. We are also planning this year to evaluate contaminant levels in embryos across Lake Erie and Ontario sites, just to see if there are any chemical influences that might impede larval development. And so once we confirm site-specific spawning, we'll start experiments that will directly test what factors are going to influence early life survival. So here's a highlight of the genetic work. This looked at the strain assignments from wild lake trout from 2008 to 2016. And what you can see on the right pie chart is that the majority of them were assigned to Seneca strain. And here on the left, you can see what was stocked. The light blue and the purple represent US and Canadian Seneca strain stocking numbers. And as you saw in O'Malley's presentation, most of the adults that were out in the lake at reproductive age were also Senecas. So this just really confirms, it links the fact that most of what we were stocking were Senecas, and most that were surviving were Senecas. And so it really, really mirrors that information. When we talk about the recent publication by Osborne et al., they were able to show that thymin levels are consistently higher in the Klondike humper strain that were stocked. And you saw in recent history that that group of fish were showing up as mature adults. And so having consistently higher thymin may help in better early life history survival. And so the high levels of thymin that they were able to observe was also linked to their diet, where Klondikes typically ate less alewife than the other strains did. Okay, for priority 4, this one focuses on population dynamics, survival, recruitment, and some strain performance. Some of the work that has gone on is we've completed a yearling post-stocking survival study, and looking at the adult catches, O'Malley and others have been working on strain performance evaluations. Next steps include a constant reevaluation of our surveys and needs to identify if we're getting the data that we're looking for, and if there's opportunities to change the surveys, as Dr. Weidel was talking about. And we want ultimately to work on some models that can estimate the density and biomass of both wild and stocked lake trout to have a better understanding of population dynamics there. And so again, a highlight of some of these projects. This is the work that Fish and Wildlife did looking at short-term survival and movement of stocked lake trout. There's a lot of information here, but essentially, survival after 35 days was high. We had about 89% survival post-stock for 23 and 24. Our treatment was different stocking depths, and so the 5 meter and 50 meter deep release groups tended to move deeper. You can see in the bottom left here. And the 100 meter group moved shallower. They all seem to be moving towards a 50 to 70 meter depth. They also had the 50 and 100 meter group had similar emigration rates. You can see in the top right, they stayed within our array for a longer period of time. But the shallower group, once they hit the water, they started leaking out of our array into other parts of the lake. And so a major finding from this is that most of the mortality that we observed occurred in the first 4 days, which is typical of a lot of stocking events. And we also had predation tags, so we knew like what proportion of these were predated on. And I don't remember what the data was on that, but it was kind of a mixed bag. It was like 50-50. Some fish were dying just from the stocking stress, and some were dying from predation. So looking at some strain-specific performance, here's some evaluation that's been going on. On the left here, we have catch curves across time and among strains. And so what we've seen here is that survival varies across strains. Some strains have better survival than others, but is fairly consistent across years within strain. And so if you have a good performing strain, it seems to perform well each year. If it's a poor performer, it consistently performs poorly. And so then if you look at the box plots on the right there, across 2008 to 2018, annual survival ranged from 25 to 91%, averaging around 69%. And you can see Klondike had the lowest annual survival rate there, and Seneca Lake had the highest. So looking at these strain differences and understanding how they perform really matters when we start thinking about future stocking strategies and how to influence our adult abundance. So finally, again, with strain performance, we looked at growth rates across strains, and what you can see here is Klondike's in yellow had the slower growth and lower max size. Lake Champlain and Seneca, as you would imagine, performed very similarly. And Perry Sound, which are just sort of showing up now, seem to be growing the fastest and have the highest max size. So that's the update I wanted to share with everybody here. If there's any questions, if we have time, I'd be happy to talk about more. And if you want more details, we can talk later. Thank you.